What if we could identify the role of a gene just by simply switching it off? What if we could then take this tool and treat diseases like cancer or AIDS just by switching off the gene that causes them? Well, in fact, with the discovery of microRNA technologies, there is hope. Scientists have uncovered that microRNAs are an abundant class of small non-coding RNAs, about 22 nucleotides in length, that play an integral role in the regulation of gene expression and the function of other protein-encoding RNA. These small molecules find RNAs with a complementary sequence, or one similar to them. They bind to them, prevent or silence these RNAs from being transformed into proteins. Susanna, Obad and her team of colleagues designed an approach to inhibit the function of microRNA and silence microRNA families by targeting tiny modified RNA-like sequences of locked nucleic acid, commonly known as LNAs. To inhibit the function of microRNA, the oligonucleotides, or the short single-stranded DNA or RNA molecules, of a sequence must be optimized for increased binding affinity and improved nuclease resistance against enzymes that come in and separate the sequences. The team synthesized these short molecules of the LNA with a phosphorothioate backbone. They then targeted the seed region, which is 2 to 8 nucleotides in length, of a series of microRNA sequences. This caused LNA to possess a higher binding affinity as well as a perfect match complementary site to the seed of the RNA. Obad and her colleagues tested their knockdown strategy in cultured cells. They took microRNA 21, a potentially oncogenic microRNA, and targeted it in the cells with the tiny LNA. From this test, the team found a perfect match for the microRNA target site. The team then designed 3 additional complementary 8 LNAs and targeted them across a series of microRNA sequences. This caused LNAs to target other regions in each sequence, indicating that no binding occurred. These findings highlight the importance of targeting the microRNA seed region to inhibit its function. The team then systemically delivered tiny LNAs in a mouse tumor model, resulting in tissue uptake and knockdown of microRNA. These findings suggest that tiny LNAs are useful for validating disease-implicated microRNA targets in animal disease models. In a recent study in 2012, Stenvang and her colleagues manipulated a similar approach to inhibit microRNA function, therapeutically targeting microRNA for treatment of hepatitis C virus, or HCV, an infection that is a leading cause of liver disease. Chimpanzees that were infected with HCV genotype 1 were treated with an LNA-modified compound termed microRNA. This led to long-lasting control of the virus. Humans with chronic HCV genotype 1 were also treated, demonstrating the compound to be well tolerated and once again providing long-lasting suppression.